Okay, can somebody confirm you can hear me?
Yes.
Thank you.
Mm-hmm.
All right. Before we begin, are there any questions? Not hearing anything, so let me share my screen. All right, you should be seeing my screen now. The lecture lesson is just finished, so that'll speed up my computer. All right, let's get out of here. So if you come to Canvas, I think it's week 5 under the lab. We have the unknown report. The unknown instructions, what I asked you guys to look at or read. I'll talk about that now. There's also a sample journal, building a dichotomous game plan, unknown species list, and the unknown notes, and an unknown grading rubric. All of this is involved in the unknown project, which you'll be doing from essentially tonight to the rest of the term, or almost the rest of the term. And then you'll submit an unknown report, which I don't have that link here, but it's down here near the bottom, which will be due November 30th. The unknown notes pages, which I'll talk about in a little bit, close November 29th. Any question about any of that? And while we're talking about the unknown project, let me go ahead and open up all the tests you can do in the unknown. Oops, too far. There it is. Week 5, lab unknown tests. All the tests you can do. Come on. On your unknown project, which kind of would be overwhelming. That's why I didn't have it shown earlier. But you won't necessarily do all of these tests. You'll only do certain of these tests, and we'll talk about the unknown project in just a little bit. All right, the unknown project is a project that we've had in microbiology from actually before I started at Clark College. It's been something that's very popular among microbiology classes. And I actually did an unknown project when I was an undergraduate. So it's been around for a long time. You're going to be identifying an unknown bacteria. In this project, the written report will be worth 75 points. It is a project in the lab. Because everything we do will be done online, what I'm going to do is send you a number, and that number is going to represent a bacteria species, which let me show you the possible species you can work with. I don't think I have that up yet. Which you can get the same file. Boy, that's taking a long time to load. You can get the same file right here. It's the unknown list or the species list. Click on that link. You can get this file here. I'm going to open it independently because it'll show much better than if I show it in Canvas. And where is that? So it'll be one of these species here. There's about 12 gram negatives and about 12 gram positives. So I'll give you a number and it'll be one of these species. There's no way to know which species it'll be, so you'll have to run different tests trying to identify the species. For example, let's say I give you, I don't know, species 111, and it happens to be Citrobacter furundi. Let me highlight that. And one of the three tests I'll give you at the start is the gram stain. And so that'll be gram negative, so that'll narrow it down to these people here. And then you can discard considering the gram positives shown here. And I think there's one more there I need to show. And then I'll give you the gelatin test. Citrobacter furundi is gelatin negative, so it'll narrow it down to this, this, and these. So these are gram negatives, gelatin negative. And then I'll give you the PR lactose test, and this will be AG for acid and gas. The possibilities for PR lactose is negative, meaning the tube remains red or pinkish. Yellowish means the tube will be A for acid, right here, or acid and gas. And I'll show you gas if you don't know. Let me see. I don't have it under here as lactose or lactase or PR glucose or PR mannitol or whatever the PR test is. It's under here as PR carbohydrate. Each of these tests is the same. The difference will be which sugar is in the tube. And so all of them will be under PR carbohydrate, and then you just look at it for what sugar it is and change the name from PR carbohydrate to either PR glucose or PR sucrose or PR lactose. Boy, that's taking a long time to show. This doesn't show. I wanted to show you a picture. I'll have to go to Google to show you a picture. Oh, there it goes. So all of these are possibilities. There are other possibilities, but these are the only ones on our table. So negative's a possibility. The tube will be pink or red. No evidence of fermentation. A, there's acid, and then Ag, acid and gas produced. The tube will be yellow for acid, and there will be a gas bubble if it's acid and gas. I've got a picture of it here. So that's a negative tube, and this is a Ag tube because the tube is yellow, and there's a gas bubble in the inverted Durham tube. Any question about any of that? And that reminds me, when we're talking about the species list here, if you don't understand something on the table, you can scroll down here and there's a key. Really extensive key, by the way. Um, there isn't AG here, but it's in the PR carbohydrate test. I'll talk more about the table in a little bit. All right, so PR lactose AG means it'll narrow it down to these four species. So we narrowed it down to 11 or 12. I don't know, narrowed it down to, I don't know, 7 to 8, and then narrowed it down to four with these three tests. I will be giving you the first three tests. After that point, you guys will be deciding which test to run. And since you've narrowed it down to these four species, in this case, you would then want to find a test to narrow it down further. This could be a possibility, so could that, but these aren't good possibilities because a V means variable, and that means the test could come back as either a positive or a negative. And that means if there's a V there, that'll fit both a positive and a negative. So a better test might be this one, not the best, or this one that'll split out one from three, meaning one positive indole positive, three negative, or this one methyl red, which will split it out to two and two, two methyl red negative, two methyl red positive, or VP, splitting out two negative, one positive, one variable. Now the variable could come as a negative or could come as a positive. And there is citrate, which you can split it down to three positive, one negative. The point is there's a lot of tests you can run to narrow it down, and you guys decide what you want to do after I give you the first three tests. The reason why I give you the first three tests is when we were first thinking of this project, when we first made microbiology online during the COVID-19 epidemic, we decided to keep the unknown project, but we were going to have communication between the student and the professor. And I looked at this and said, well, geez, on average, the student's going to need to request, I don't know, something like 5 or 6 tests. There's 20 different students in the class. That's at least 100 different communications for one project for one class. And I decided that was too many for, did I do that right? Each student, there's 20 students, and there's 5 tests. Yeah, 100 different communications. That was too much for one project. And so I asked them, how about if I decide the first 2 or 3 tests, and then the students run the next test? And they said, that would be really good because that will cut down the communication between the professor and the students for this one project. Any question about any of that?
I do. You said you're going to email us
Yeah, I will
that.
email you the results to these 3 tests, the gram stain, the gelatin hydrolysis, and the PR lactose test. The reason for the gram stain is it's very simple, dividing the possibilities down in half, gram negative and gram positive. The gelatin test I decided to run because this is the test that takes the longest. This test will take a week before you can get the results. So if you were to ask for a test, you won't get the gelatin test for a week after you ask for it. And you can look up how long each test takes. Gelatin is the longest. Come up here. PR carbohydrate test, you can get it within 24 hours and at most 48 hours, which it'll tell you up here how long it takes to incubate. There you go. Should be incubated for no more than 48 to 72 hours, and ideally, for 24 hours or less. I think ideally it's 18 hours. Yeah, 18 to 48 hours right there is the ideal time. Beyond 48 hours, you may get problems. So on this one, I'll run it either at 18 hours or 48 hours, and most 48 hours.
And when you
Question
say
about
ask, you mean like email you and ask you?
no, everything except for the very first email, which I will email you your number, which will stand for your species, and then your first test results, which will be the PR gram, no, the gram stain, the gelatin hydrolysis test, and the PR lactose. I will email that to you in Canvas, and then you will take that information and put it in your unknown notes pages. Let's go to that now, since the question is, how do you work this? Click on the unknown notes page, and what I have to do is, let me stop the share here for a minute. I'm going to go to an unknown notes page from a previous term so I can show you what the page actually looks like. But I got to remove the student's grade, so I can't show you the student's grade. Um, Dashboard, take a previous term. So you're not even going to see the term. I'm not going to mention it. And I won't mention the student's name, although you can barely read that. But, that's not the best way to get there, but read it here now. You're not seeing this, right?
No.
Great. Okay, let me start sharing it now. So you will put your results, your conclusions, what species are under consideration, and anything else about your unknown project, you will put them in your unknown notes page. So on February 6th, the student said they were starting the test, gel hydrolysis test. February 10th, they started the gram test. On February 11th, they started the PR lactose test. And then on the 13th, they got the results from all of these tests. And here's the results of the gram stain. And what's their conclusion? Gram negatives. There's the results of the gelatin hydrolysis test. The bottom tube is a positive gelatin hydrolysis. And then there's the results of the
Sorry, we
lactose
could,
test.
sorry to interrupt, but we can see their name and grade.
No, you can't see the grade. I've got it erased. I
Uh,
can't remove the name. Sorry.
oh,
I can
you're
only show
like,
you. I
I
can only
just,
show you what the unknown notes page is supposed to look like.
okay. It says at the top bar, like the average. Is that just everybody's grade? Sorry.
At the very top, where is that?
Okay.
Where is that?
Like right beneath, it says like 14.08
Oh,
out of,
right here?
yeah, is that
Um,
just the
I'm
average
not
of,
sure what that is.
okay.
15 out of 16 are graded. All of them should be graded because this is a previous term. And this is 4 out of 16. And I don't know what that is. This isn't the student's grade. The student's grade will be
Okay.
right here. Okay. This might be the class average. I don't know.
Okay, thank you. Sorry, just wanting to make sure we didn't see it.
No, you're not seeing the grade. Like I said, I've got it erased. It's here. And this is the PR lactose test, which you can see the results there. and the tube remained red, indicating a negative test result. So the conclusion is negative. Uh, and then the result is it's a red tube. Now, let me state that here you will be graded on stating the result and the conclusion. And the result is what you see. The tube is red. The conclusion is, what does that mean? This test is negative. The PR lactose test is negative. Okay, so understand the result is an observation and the conclusion is mainly whether the test is, in this case, negative or acid or acid and gas. That's the best one to show it here. This one, the tube is liquid, meaning you tilt it, it pours, and that one's solid. And this is the tube, so this would be positive for the gelatin hydrolysis. But the result will be the tube is liquid. And here the cells are pink, and we got to blow that up to see it. I can't blow it up any bigger. Blow it up here, that worked. And you can see there are pink rods. Ah, come on. Pink rods. That is the result. The conclusion is they're gram negative. Any question about any of that? All right, let's shrink this down. Anyways, the point is the very first time I will send you the results, you need to put the results in your unknown notes pages, and then you must make conclusions, put that in your unknown notes page. You should put the species under consideration. Let me see if he's got that here. Yeah, right there. So he's got the species under consideration right there. And it looks like those are under consideration as well, because he's talking about the next test. And then you ask for the test. You put it at the end of the unknown notes page, the test you're asking for, and then I'll see it. If you put it at the start of the unknown notes page, I will see it. But if you put it someplace in the middle of the unknown notes page, I will not see it until I actually read the page. And initially, I will be looking at the unknown notes page about every other day. The exception would be on the weekend. I may not see it on Saturday and Sunday, meaning I will see it on Friday and Monday. But on every other day, I will be looking at it about every other day, sometimes every day. And I will only be looking at the start of the page, because that's the page that loads up. And then I'll scroll to the bottom of the page to look for your test request. And if you have your test request at the bottom of the page or the start of the page, I will take your test request. If it's in the middle of the page, so don't put it in the middle, I will not see your test request until I actually read your page. And I will not be reading your pages every other day. Okay, any question about any of that? Meaning I'll just look to see what the test request is, and if it's time, meaning the test has to incubate after the incubation period, I'll then send you the results. I'll put the results in the comments. So don't use the comments. Put any comments you want in the note page, your unknown notes page. Put it in here. I will find the comment. This student apparently put a comment there because I put a note in here, don't use the comments. And after a while, he put the comment in the unknown notes page and then I deleted it. I will delete your comments in the comment section. The comment section is for me to send you comments, and I will send you the different test results. Like the first test he asked for was the indole test. And after this test incubates, I will then send you the results of the indel test and the comments. Okay, any question about any of that? So in your unknown notes page, do not use the comments section. It's for me. And another reason for not using the comments section is Canvas does not warn me or flag it when you've made a comment. So I may not see your comment if you put it in a comment section for days, weeks. There's even been a comment made that I found uh, like a term after the term ended, meaning I found it in the next term. There was a comment, somebody put it in the comment section, and I never knew there was a comment there. So don't put your comment in the comment. What you want to do is, if you have a comment, put it in your unknown notes page. I will read it. Any question about any of that?
Yes, not about the comments, but that it can be a working document that we can go back and edit even if we like, because it just says submit the assignment, but it's something that we can go back and fix and add to.
You can add to it. This student has 1, 2, 3, 4, 5 different pages. You can make actually an infinite number of pages here, but let's see what he's done. On this one here, he is adding to it, like on March 5th, he has results up until February 28th. And so on this one here, I'm not sure why, he didn't submit it initially in February. Oh yeah, he did. Sorry. February 13th, what did he have here? I don't see anything. Looks like he submitted a blank document for whatever reason. Oh no, it's just taking a long time for some reason. So we've got here just what the tests were. The first test results. And then on February 5th, he asked for, on this day, he asked for the end-all test. And on February 5th, no, February 16th, he put up the results of the end-all test. And in this case, I'm pretty sure he added to it. I am not going to go to another page because you can see how long this takes to load. So what he did was he just put the end all at the bottom of the document. You don't need to do that. Put it at the bottom of the document, meaning he's got the previous document earlier. But if you do, it will make it easier for you to write your unknown report, because if you write the unknown notes formally, you can just copy and paste the unknown notes and then put it into your unknown report. Now, the unknown report will contain more things than the unknown notes, but you can get started on your unknown report just by copying it from your unknown notes page. And I suppose if you were to do it with each day having a report, you could just copy it 1, 2, 3, 4, 4 different times and put it into your unknown report. So you can do it either way. If you write your unknown notes formally, then you can just copy and put it into your unknown report. If you do it informally, meaning you're not writing with good grammar and not being formal, where you give the date and state everything you should. Of course, that has to be in there, or you'll be graded down. But I don't know, informally, you're talking like in slang and stuff like that. Um, you wouldn't want to put that in your unknown report. You want to only copy and paste it in your unknown report if you're making your unknown notes formally. The unknown notes page can be informally, but if you do it formally, it'll be easier to write your unknown report because you can just copy and paste it. Any question about any of that? All right. The unknown notes page just has to have, actually, I think I got it stated up here. Record your daily activities for your unknown project. Everything will be recorded in the unknown project in your unknown notes page. That will make it easy for both you and I. If we want to look up something, we know where to look for it. We look in the unknown notes page. If, on the other hand, this were to be done as a bunch of emails, and I were trying to look up something, it would just be a mess. I would never find it because I get so many emails from 20 different students. And even for you guys, it'd be much more difficult to try and look up something in an email. But if it's in the unknown notes page, it'll be easy for both of us to look it up. Put your test results here. Put the test request, your next text request, at the bottom of the page or at the top of the page. And then I'll provide the test results. That'll be in the comment section. You need to record the results of your test, state what the conclusions are, and then give the species under consideration. Let me make sure that's stated up here. Yep, right there. As well as the species that are under consideration. All right. And I give you a warning, if you put things in a comment, I will delete the comments. Okay, I'll give you a warning. I'm going to delete this comment. Move it to your unknown notes pages. Don't use the comments. All right, anything about the unknown notes pages? All right, we talked about the species, how we're going to do it, and I'll send you a number. It'll represent the species. There's no way for you to know what species it'll be from the number. The only way you'll be able to figure it out is by running different tests. And your test will be to identify the unknown species. Let's go to the, let's go ahead and close that down now. Oh, let me shut this down. I got to put the student's grade back. It probably won't be a problem, but if the student were to contact me in the future asking about his grade, I want to have it here. So let me store that, and you're not seeing this. I put in his grade. And let me share my screen now. All right, let's talk about the unknown project. You can read about the introduction and the rationale. The project format, like I said, everything will be online. I will send you a number representing a species, and then I'll send you the first three test results. After that point, you put everything in your unknown notes, take a look at the results and the conclusions, figure out what your species possibilities could be, and then decide which test to run. And the tests are up here. The species are here. Decide on a test, and then read about the test, find out how long the incubation period is. Most of these tests have 2 days incubation, meaning if you request a test, I'll start it, and then after 2 days, I will send you the results. And I'll send you a picture of the results. Some of the tests you can get back within the same day or a day. The catalase, the oxidase test. There's one other test, I don't remember what it is. There's 3 tests that you can get back within a day. Pigment test, there it is there. Within a day or less. Okay. Um, the exception would be like oxidase. If your cells are very old, I will require you to grow new cells, and that'll take a day. But you should get the results within a day. Um, Most of the tests take a day, I mean 2 days, like the urease test, sodium chloride test, hemolysis test, the indole test. A few tests take longer. The methyl red test takes 5 days. Okay, and you can look up on the test how long they take, but most of them are 2 days. Um, we're done there. So you're virtually going to be receiving a broth culture, and you should streak it out. You're going to virtually streak it out, meaning you're just going to pretend to do all of this, and you'll streak it out for colony isolation, get an isolated colony, streak it out a second time for isolated colonies, take the cells from that colony to set up your stock culture. And then from your stock culture, you will uh, pretend to set up all your future tests. In reality, you will put in your unknown notes page the test you're requesting, and then I'll set it up. And then after the incubation period, I will send you a photograph of the test result. You will then put your results in the unknown notes page. You will make conclusions, put those in the unknown notes page, and then put the species under consideration. Put that in your unknown notes page. And then at the end, put your next text request. Put it at the end of the page. For the tests, you can use your lab textbook, the lab manual, especially if you have the photographic atlas. And then you can go online to look up test results. You can find Berge's manual either in the library or parts of it online. And then there's my own page in Canvas, which sometimes shows you the pictures of the tests. I don't have it here anymore, but I used to have the PR glucose, and I showed you the picture of that. And then you should look under there. Let's come back here. You should look up at the test when you request the test to see how long that test will be before you get the results. As well as there's pictures about what to look at. I think with the capsule stain, I've got pictures. Now this will take a while to load. So while it's loading, that one. Let's talk a little bit further. You will be graded on your logic that you use to perform the unknown project. You should be using dichotomous logic. Dichotomous logic means taking one test, having a yes/no answer. Let's scroll down to this page here. Initially, we did the gram stain, and it was either gram positive or gram negative. Gram positive are shown here, all of these species. We then did the catalase test, and the catalase positive are shown here. The catalase negative are not written, so these others would be catalase negative. And the result was catalase positive. And then you ask for the PR mannitol test. It's PR mannitol negative, so it's these three possibilities. Staphylococcus aureus was thrown out. PR glucose, you get an acid result. So these two are possibilities. Micrococcus luteus was thrown out. And then citrate positive means that we knock out Staphylococcus epidermidis and are unknown as Micrococcus varians. This is following dichotomous logic where you're using one test at a time, setting up your next test, getting your results, etc. That's dichotomous logic, where, let me show it here. Well, if I can get it, let's try that again. There it goes. I don't know why that changes. All species, for example. Oops, that's the wrong thing. And then we do the gram stain. So there can be gram positive and gram negative. I'm just going to abbreviate that positive for gram positive. We'll go capital G, positive. Yep. Help if I could type correctly. And then the others will be gram negative. And let's say it's not gram negative. Cross that out. And then we do the Cadley's test. Cadley's positive. Let's just put that Cadley's next. Yep. So there's positive here. Cadley's positive. And Cadley's negative. See how the dichotomous logic works? Now we're going to cross out the Cadley's negative. And so we have the, I don't know, we're going to do, let's just say oxidase test. So there's oxidase positive. And oxidase negative. All right, so we start with all species, narrowed it down to the gram positives, narrowed it down to Cadley's positive, gram positive, and then if we cross out the oxidase negative, it ends up with oxidase positive, gram positive, Cadley's positive, sorry. Oxidase positive, Cadley's positive, gram positive. Which I think there is one that way, and it's Micrococcus luteus. So I've identified the species. All right. The point is you must use dichotomous logic. If you don't use dichotomous logic, you'll get a penalty. Shotgun logic is like when you're taking a shotgun, you're just shooting, hoping to hit something, and you could hit lots of things, for example. Should not be used and result in a stiff penalty. You should write your daily lab notes in your, what do I call that? Your unknown notes pages. And then write your unknown report. The unknown report will be worth 5 points. The unknown notes pages will be worth 5 points. They will be graded, but they will only be worth 5 points. The unknown report will be worth 75 points. In the unknown report, you must submit a report that details your thoughts and activities for each day of your unknown work. Please use 12-point font. I've got glasses, so if you make something smaller than 12-point font, I'll have a difficult time reading it. Do not double space your pages. You should have your daily description, meaning what test you ran, what the results were, what your conclusions were you made, what the species were under consideration. And then probably at the end of that, you give, I think I'll run the next test, which will be this. Do not omit data to make your data look better. Drawings, photos, and tables can be very helpful. They are not required, but you may include them in your unknown report. Drawings, photos, and tables must be completely labeled and discussed within the body of the report, meaning if you get a photograph from the web, you have to state where you got that photograph from. I guess it would be an exception, and that is if I send you the photograph and then you put that in for your results, you don't need to state where you got that from because I know I got that from, you got that from me. Photographs, drawings, tables are not required, but they are very helpful. You must report each entry with the date. You don't need to have a time here. The time was for when we were, oh, I don't have the unknown notes page up anymore, but it will assign a date to it. Let's go on to here. You won't have a Spree grader page. You'll have something like an upload page. And there's nothing here. So if you do, there will be a date here. And you just need to put that in your unknown report where the date is. And just get it from your unknown notes page. The time was for when we were actually coming into the microbiology lab, and some students, especially near the end of the term, would come into my lab and another instructor's lab on the same day. And that I allowed as long as they only did one test in each lab, meaning each, what do you call that, course day. And that's following dichotomous logic, but you won't have that as an option. So you will have at most probably one test on each day, but you'll be doing it online. So you can do it Monday, Tuesday, Wednesday, and Thursday, for example. And the students in the class, they could only do it on generally Tuesday and Thursday, but they could come in an earlier instructor's lab as well as my lab. And then on Thursday, they could come in the other instructor's lab and then my lab. So they could come in like 4 times a week. But you don't need to have a time, you just need to have a date. You do not need to describe the step-by-step details of each test you run. For example, if you run the gram stain, all you need to do is say, I asked for the gram stain. Here's the results of the gram stain. There's the conclusion. You do not need to have a step-by-step process of what you do in the gram stain. I know how to run the gram stain. You should give your thoughts and your words in your report. They should be your own. If ever they're not your own words, more than 3, you should put them in quotes and say where you got it from. This is especially important for those of you who are using artificial intelligence to help write your report. You should state where you got the information from artificial intelligence. Like if you're using Grammarly, all I want you to do is state, I use Grammarly, and this is what I grabbed from Grammarly, okay? And you can put it in quotes, or you can, if it's like, I don't know, several pages, paragraphs long, I guess you should put it in quotes and then state where you got it from. And I do require you to do that if you're using artificial intelligence. I just want to keep track of how frequently artificial intelligence is used in microbiology. It's not anything you'll be graded on, I'm allowing you to use artificial intelligence, but I do require you let me know. And like I said, I can send your report to artificial intelligence and ask it, was artificial intelligence used in writing any of this report? And it will tell me what was used and probably which artificial intelligence used it as well. So do state it. If you don't, I will mark you down for plagiarism because you're using artificial intelligence and it's not your own words. And even if you change artificial intelligence, the wording to your own words, I still want to know about it. So you should, well, you probably put it in quotes because it's your own word, but you should state that the above paragraph was my wording and came from artificial intelligence and say which artificial intelligence. Like I said, I want to keep track of how frequently artificial intelligence is used in microbiology. All right, it's mandatory that your report include a table or a figure outlining the process you use in identifying your unknown organism. An example would be initially the student did the gram stain, narrowed it down to these species. Then they did the catalase test, narrowed it down to these species. Did the PR mannitol, narrowed it down to these species. Did the PR glucose, narrowed it down to these two species. Did the sodium citrate test, narrowed it down to Micrococcus varians. And now they've identified the species. In the project, you may do one confirmation test, meaning after you've identified your species, you may do one more test just to make sure you've got the correct species. And with Micrococcus varians, what would be a good test to run? Well, if you wanted to separate these two, you could do oxidase test. If you wanted to separate these two again, what would be a good one to do? Oh, you could do the a pigment test, separate these two. So here would be a good one to do the pigment test to make sure it's Micrococcus varians. If you don't know, Micrococcus varians is yellowish in color. So you're allowed to do one additional test. You won't be graded down for a confirmation test. However, if you do two additional tests or more after you've identified the species, you will be marked down for using excessive media. If you do not finish the project, sometimes students wait to the very end before beginning the project. In which case you don't finish the project, just write it out as far as you can. Let's say you do the PR glucose test and you narrowed it down to these two species. All right, you have to write your report on that. I've narrowed it down to these two species. I need to do the sodium citrate test. And if it's positive, it's Micrococcus varians. If it's negative, it's Staphylococcus epidermidis. But you can't identify the species. It's one of these two. Um, Hopefully you won't wait to the very end. You should start this project soon because actually you have to wait for me, and the first test will be a week from tonight. But once I give you the results, you should start the project soon because this project will take time. On average, you will need to run 3 or 4 tests after I've given you the first 3 test results. Now, there's some very lucky students, maybe one student a term or one student every other term. All they have to do is ask for one test, and if it's the right test, they can identify their species. But that's a very unusual case. Most students will need to run 3 or 4 tests. An unlucky student will need to run 5 tests before they identify their species. 1, 2, 3, 4. So this is a total of 5 right here. So get started because this project will take time. When you request a test, it will take time to incubate. Like the catalase test, you can get that a day later. The PR mannitol, you can get that between 1 day and 2 days later. Same with PR glucose. The sodium citrate test will take 2 days to incubate. And if you ask for the sodium citrate test at the very end, and the report is due before the citrate test incubates, you're out of luck. You're not going to be able to get the results. Any question about any of that? All right, so it's required you have a table or a figure outlining how you identified your species. If you have a test that doesn't help you narrow the results, you don't need to put it in the table or the figure. Like, let's say you did the gelatin test, and it gave you the same results as the catalase test. So that doesn't help you. You don't need to put that in the table or the figure. You do need to put it in your report, but you don't need to have it because you didn't use it for identifying your species. It's also mandatory that you have a paragraph at the end of your report talking about the microorganism you've identified. It only needs to be a paragraph, meaning a couple of sentences discussing the species Micrococcus varians here. And no longer than a half a page. So only a paragraph or two in length. Tell me about your microorganism, um, including any diseases it causes, modes of transmission, common places it's found, treatments, any other interesting information about it. Just a paragraph or two. You do need to give a reference for where you got your information for the microorganism paragraph. So you must have also a reference section in the report, and that'll be at the very end of the report. You may only have one reference, okay? If you only had one reference for writing your microorganism paragraph, that's all you need. If you got your information from more than one source, then you have several references. The references can be in any appropriate citation style, such as APA or MLA. All right, I want to remind you plagiarism is not allowed, so don't plagiarize. If I detect plagiarism, you'll get at least a 5% penalty, and if it's egregious plagiarism, you get 0 points out of 75. The grading criteria, how I'll grade the unknown report, is here. You'll be mainly graded on the logic you use in performing your unknown report. Thought I had it here. You'll be graded on the completeness of the report. Each entry should include as much detail as possible. What tests were run? What were the results? What were the conclusions? What species were under consideration? Completeness and accuracy count. You'll be graded in organization, clarity, and neatness. Each entry should be dated, and then you should talk about each entry on that date. And normally on a date, when you're in the lab, you will have a result from a previous request on that date, and you'll give your results to that date, your observations, you give your conclusions. You'll then discuss the species under consideration, and then lastly, you will give a future test request. So that would be part of your organization. You'll be graded on written communication. If there's no grammatical or spelling errors, you'll get a perfect score on that. If the paper is easy to read, easy to understand, you'll get a perfect score on that. Successful identification of unknown is worth, I don't know, 5 or 10 points. Let me look that up. And I've got the grading. This last part of the unknown project goes through working on a dichotomous logic. If you're not familiar with dichotomous logic, you can read the last part of your unknown report and you can work the questions right here. You do not need to turn these questions in. You do not, if you're comfortable with dichotomous logic, you do not need to even read this part of the unknown report. All right, let's go to the grading. See if I have that. I think this I can shut down. There it is, unknown report key. Out of 75. If you get the correct species, you'll get 10 points. If you get the species within the same group, you get 7 out of 10. But the gram stain has to be correct for 7 out of 10. If you're outside the group and the gram stain is correct, you get 5 points. And then if you get the wrong gram stain, you get 0 out of 10. So if you identify the correct species, 10 out of 10. And let's start at the top now. Utilization of media. Was any excess media used or irrelevant test performed? If there's none, then you get 10 out of 10. If you have one test that didn't help you at all, or it was an irrelevant test, you're using excess media, and you'll get 8 out of 10. If you have two irrelevant tests, you get 6 out of 10. Was dichotomous logic used? Meaning there's no excess tests, you get 10 out of 10. If there's one excess test where you're asking for two tests at the same time, you get 7 out of 10. Two excess tests, meaning you're asking for three tests at the same time, you're getting 5 out of 10. If you use shotgun approach, you're just asking for a whole bunch of tests at one time, you get 3 out of 10. No logic used at all, 0 out of 10. Were all relevant tests used? Generally, students get 10 out of 10. The exception is if a student runs out of time and they need to run one more test to get their species identified, they'll get 8 out of 10. If two tests needed to be run, they'll get 6 out of 10. I've never had a student require more than uh, two tests to get the project finished. Were correct species included at each step? All species given were correct. If one species is incorrect anywhere in your test, you get 8 out of 10. 2 to 3 species incorrect, 6 out of 10. Over 5 incorrect species, or if you do not state what the species under consideration are, you get 2 out of 10. Actually, if you don't have any species, it'll be 0 out of 10. Complete list of report, all the information presented. One instance of missing information, 8 out of 10. Like if you don't have your paragraph discussing your bacteria, you'll get 8 out of 10. If you don't have your paragraph discussing your bacteria and you don't have a reference, you get 6 out of 10. Okay, something else is missing. Actually, two other things missing, you get 4 out of 10. I don't know what would be missing, but if something I found was missing, you'll get 4 out of 10. Effective communication, was your English, grammar, spelling, and punctuation all good? You get 15 out of 15. If you have a few errors, but I can understand your logic, you get 13 out of 15. If you have some errors and some of your errors, I can't understand what's going on, don't understand your logic, you get 7 out of 15. Any question about the grading? All right. Now I have a bunch of things on my Canvas web page to help you write the report. There's a sample journal, what your unknown report should look like. Oh, let's not show it here. Let's go here. This journal was actually written by an instructor about identification of unknown number 6. Please give your number. I'll mark you down if I have to look up the number. By who, when, obviously you won't do it in 2004. You should put your class name there. Initially, they streaked out the culture and they had a mixed culture and streaked it out. That's why they've got two different colonies here, one yellow and one white. You're going to be only given one number, which will represent one culture, meaning you're going to get a pure culture to begin with, but I do want you to pretend to streak it out and just write in your report, I got the unknown number on, well, actually, it's going to be tonight, assuming I send it to you tonight. And I took the unknown culture and streaked it out on a TSA plate or a nutrient auger plate, streaked it out for isolated colonies, picked an isolated colony, streaked it out a second time, got isolated colonies, then took those cells from one colony to set up my stock culture. Ran other tests from those stock culture. Let's come down here. He's showing you the results of the gram stain here, which it's hard to see this. Let's see if I can blow that way up. All right, that looks like a cockeye. It's obviously purple. So you can say the cells are purple cockeye, and that means they're gram. The conclusion, that's the result, purple cockeye. And the conclusion is they're gram positive cockeye. Do distinguish between your results, that's an observation, and your conclusion, that's an interpretation. So let's start on this day here. He's got the date, the observations on the stock culture. You'd only really do that if you're looking for pigmentation. And the activities, he did the gram stain. And here are the results of the gram stain. And there's the conclusions of the gram stain. And then you give your next test results quest here. He doesn't have that there, but he's got it here. He's going to do the catalase test on the next day. So that's the activities done. There's no observations for today. And then the conclusions on the test, the results were he saw bubbles arise from the catalase test. The conclusion is these cells are catalase positive. And then he's got the species under consideration. The species is either a member of Staphylococcus, either Staphylococcus epidermidis, Staphylococcus aureus. I don't know if there's another Staphylococcus, or a Micrococcus species. And there's at least 3 Micrococcus species. Micrococcus luteus, Micrococcus varians, and what's the other one? Don't remember. Now there's a 3rd Micrococcus species. Anyways, this is what your report should look like. Here he's got his table here. He narrowed it down to Staphylococcus aureus because that ran in each of these tests. Normally, you would have this is a possibility, the gram positive, the pigment, these three species, catalase positive. You should really only have these three species. S. epidermidis is out of the running because it wasn't pigmented. And then beta hemolysis, Staphylococcus aureus. So in reality, with this one, he's now identified the species. And then he did the biolisculin, mannitol, and narrowed it down to species. He did 2 confirmation tests. You are not allowed to do 2 confirmation tests. If you do, you'll get a penalty. You should only do 1 confirmation test, and you don't have to do a confirmation test at all. Once you've identified the species, if you're perfectly happy with it, you can stop right there. But you are allowed to do 1 confirmation test, and actually, this is not a good confirmation test because you want to do one that's catalase positive. So none of these tests are terribly great. Um, and then you got the paragraph discussing the bacteria, and then the references. Any question about any of this project? I think that's it. So let me remind you, you got a sample journal here. There's the instructions. Oh, if you want some help on building a dichotomous game plan, you can take a look at this page. Essentially, to follow a dichotomous game plan, all you need to do is select one test, ask for it, get the results, and then select your next test. So if you do one test at a time, you'll be following dichotomous logic. Now, initially, when I'm sending you 3 tests, we are following dichotomous logic because tonight you'll start the gelatin hydrolysis test. And then on is it Sunday? On Sunday, you're going to start the gram stain. And you're not going to look at the results of the gram stain on Sunday. You're not going to look at it on Monday. You're going to look at your results on Tuesday. And that's a possibility where you make the slide and then you just never look at it. And that's what I'm assuming you're doing. And then on Monday, you're going to start the PR lactose test. And then I'll send you the results to all 3 together. The purpose for that is I can send 20 emails, 1 to each student, instead of sending out 60 emails. And it just is much reducing the paperwork on my end. Like I said, the purpose of sending out 3 tests together, and I choose the first test, is largely just to reduce the amount of communication between me and each student. Having 100 different communications for one project was way too many. And I wanted to reduce that. And by doing the first 3, I'm going to reduce the communications by 60. So instead of 60, I'm going to do it 1. And that means I'll still have 40, but 40 is something I can do for one project. And then there's the species of under consideration, and there's for your unknown notes page. Once again, there's the grading rubric. You should take a look at the rubric when it's time to start writing the report, and that's probably when you should look at the sample journal as well. This project is not due until I think it's November 30th. Remember right? Yes. The unknown notes page will close on November 29th. That is a Friday, November 30th. is Saturday. So at 11:59 p.m., the unknown project closes. The unknown notes page, I close it a day earlier. And if you ask for a test on Friday, it has to be a test that I can get you in one day. If you ask for a test that takes 2 days, you're out of luck. You're not going to get the results in time to submit your project. Any question about any of that?
For the notes on the timeline, for in terms of the three tests that you're going to give us, you said tonight would be technically the gelatin test is performed, correct?
Yes,
And then
and it
Sunday,
takes a week.
okay, perfect. And then Sunday, the lactose test would be
No,
performed
Sunday the
at
gram stain will be started, and Monday
grams.
the PR lactose test will be started.
Monday, you said was the lactose test.
Yes,
Thank you so much.
PR lactose. All right. Any other questions? Let me state that this project does get some students confused. If you get confused, send me an email. Tell me what you're confused about, and I will help you. And don't send me an email saying, I'm confused about this project. I need help. Tell me everything. Okay? You got to tell me what you need help on. Okay? If you tell me I'm confused on this project, I'm going to just refer you back to this lab session because I don't know where to begin. And I don't want to go through, how long have we been? We've been talking about this for almost 2 hours, 1 hour. No, it's 6:30. So almost an hour and a half. Any questions? I think I'm done. Hopefully I'll get the project ready to send out tonight so you can get started with the gelatin hydrolysis test. However, I'm not going to send you the number or the test results until next Tuesday. The gelatin hydrolysis test takes 1 week, and that's why you'll get the results on Tuesday. And then the gram stain, you're going to start it on Sunday, but you're not going to look at the results until Tuesday. And then the PR lactose test takes a day to incubate. So that's why we're starting on a Monday, and you'll get the results on Tuesday. Any question about any of that? All right, I think we're done. I will see you in the next class. Goodbye.